Before we move on to see actually the slides from the histology guide, in the beginning I want you to see exactly the ideal examples of what is what. So in the beginning we're going to find this is the cortex and this is the medulla, and it's pretty easy to identify because we're going to find the big typical characteristic of the glomerulus. Now these empty spaces of the Bowman's capsule are very nice parts, and it actually assists us in identifying where is what. So this is again the cortex, and this is the juxtamedullary nephrons right here, and this is of course the medulla as it is. So when we zoom in the cortex, we're going to see the typical representation of the glomerulus, of the renal corpuscle, and this is exactly what we're going to see macroscopically. Now, I'll be honest with you, it's not easy to identify, of course, it's the specific cells of the podocytes, endothelial, and the mesangial cells, and no one will actually will ask you to, and that's not the point of this examination, this presentation. Now, if we're gonna find the easy things to identify, of course, will be the macula densa right here, exactly this very nice part that we're gonna see and talk about specifically again later on. Now, this is the very nice and typical presentation of the proximal convoluted tubule. Again, this is the picture that will help you understand exactly the similarities and the differences. And this is, of course, the vascular pole, where the capillary actually, the afferent arteriole comes, and you can actually see the red blood cells present in this area. And this is the tubular pole, and again, you can actually easily identify the beginnings of the proximal convoluted tubule right here based on these cuboidal cells of the proximal convoluted tubule. So this is again a slide that actually shows you specifically, these pictures are again taken from Histology Guide. I specifically found areas that are ideal and representative of the specific different parts of the duct, of the tubules. So the PCT tends to have this pyramidally shaped, let's say, areas in pyramidally shaped morphology. This is one feature that will help you identify what is what. The thin Henle, the thin loop of Henle, of the part of the thin loop of Henle, are actually, as the name suggests, thin, meaning that we're going to have simple squamous epithelia present in the circles, and it's going to be very easy to identify for two reasons: based on the location in the medulla and based on the morphology, which is of course a very nice simple squamous epithelium. Now the DCT, distal convoluted tubule, will be present again in the cortex, and we're going to have a bit more, let's say, a bit shorter cells, just a bit shorter in comparison to the PCT, and more, let's say, of equal height. And lastly, we're going to have the collecting duct. Again, this is the last part of before we actually, let's say, dispose the urine in the calyxes. Now, this collecting duct are going to have typical and very nice clear nuclei of simple cuboidal epithelia, and these cells are going to be called principal cells because they react to specific hormones. We'll see them in just a bit, which hormones work with that. So this is exactly what the morphology of the different parts of the different renal tubule parts looks like, and this is the very easy way to first identify what is which part of the tubule is which, let's say the proximal or distal and so on. And after you understand exactly how they look like, we'll discuss about the function in just a bit. So as I said before, this is the macula densa. The question is, since it's just a part of the distal convoluted tubule, why would it have a special name? Because typically in 99% of all the cases, of all the cells, all the structures that have a special name, they typically tend to have a special function. And in this case, it could not be more true. And this is because the macula densa and the juxtaglomerular cells form the juxtaglomerular apparatus. Now, what exactly is this structure? This is the structure that controls the blood pressure of the afferent and efferent arteriole. This controls the local amount of blood that goes from the arteriole to from the afferent part to the efferent part of the arterioles. So again, the juxtaglomerular cells are of course the smooth cells of the endothelia and have specific function. One, of course, is the presence of that itself actually consists a smooth muscle, which is, of course, as a consequence that it has the contraction function. And second, and most importantly, they produce renin. Now, renin is a very important molecule that plays a role in this whole renin-angiotensin-aldosterone system. Now, this is a very complex system. We'll discuss it in just a bit. And the logic is, of course, here, the point of this mechanism is to control the amount of water and ions that are present in the blood and as a consequence control the pressure of the blood, because the more component, the more water you have, the more ions you have within the tube, within the practically tube, within the vascular system, this of course has as a consequence the increased pressure. The lower amount you have, of course, the lower pressure you should have, just based on the content of liquid to tube, just mechanically speaking, because in real life, in the real human body, there's of course compensatory mechanisms that are in play when we have small amount of blood, of course, and high amount of blood, in order to compensate for this this difference in the pressure. Now the rationale is just that. So this is the first part of the juxtaglomerular apparatus, and the second is the macula densa. Now the macula densa, as again, is let's say the point of pressure of control in this case, and is a sensory organ, sensory point of the distal convoluted tubule. And these two components together play the biggest role in the formation, in the regulation of the glomerular filtration rate. Now, what is the glomerular filtration rate? As the name suggests, is the speed by which the glomerulus filters the blood. Now, for you to have an idea of how much blood does the kidney actually filter in the day, in the 3 liters that are present in the blood plasma, now these 3 liters pass by the glomerulus 60 times a day. So this is a very effective mechanism that filters blood continuously and non-stop. So now that we discussed all the different components of renal tubules, we discussed about how the PCT looks like, how the DCT looks like, how the thin segments of Henle's loop actually look like, the whole structure of the renal corpuscle. Now it's the ideal moment to start talking about the functions of the renal tubule. So the PCT, the proximal convoluted tubule, is the area that is the location in which we have the most effective reabsorption of water, of nutrients, of electrolytes, and proteins, of course. Now, these cells actually play the biggest role in the biggest proportion of reabsorption of ions and nutrients and water is exactly taking place in the PCT. This is important, and you should really have it in your head. Now, the second one is the second segment, of course, is the thin and thick limbs of a loop of Henle. Now, the loop of Henle is a mechanism that actually plays a bigger role in the countercurrent multiplication, and this is of course a specific mechanism that you should best understand and best talk with in your physiology course. This is just a mentioning of why you should really understand and really care about the thin loop of the loop of Henle. So this is the area where we have the passive reabsorption of sodium and chloride, and of course the active reabsorption in different components. Typically, of course, it's very logic that the thin segment that only contains the simple squamous epithelium will not be able to facilitate the active reabsorption. Why? Well, because in order to create these pumps, to create these ports, you need to have extensive presence of the cellular components, the RER, and so on and so on. Of course, in this case, it's really logic to understand that the active reabsorption part takes place in the thick ascending limb of the loop of Henle. So let's talk about a bit the DCT. Now, the DCT, of course, is the distal convoluted tubule, and I want you to think of the DCT as 2 different segments: the early DCT and the late distal DCT. Now, the early one actually is nothing more than just the location of reabsorption of electrolytes, again sodium, chloride, potassium, and so on. Now, the distal DCT is where we start to have this very sensitive mechanism of hormonal regulation of the blood pressure. So the distal DCT, along with the collecting system, should be thought as one unit functionally speaking. This is because we're going to start seeing the effects of aldosterone. We're going to start seeing the effects of the antidiuretic hormone in these areas. Let's finish with the system, with the collecting system, then let's talk about specifically about the hormonal regulation. So the collecting system actually is composed of 2 different cells, the principal and the intercalated. The principal cells are responsible for the reabsorption of water and electrolytes and the regulation of potassium. And keep in mind that potassium is actually really important when it comes to the reaction. It's not just any ion. This is an ion that has specifically cellular effects in this whole cellular effects, and the balance of potassium in the blood and inside the cells has to be strictly controlled. And then we have the intercalated cells that have a more reabsorptive function, and importantly, they help maintain acid-base balance. Practically means that it actually controls the amount of proton H plus ions that go in and out of the filtrate and the bloodstream, and of course, as a consequence, and also the bicarbonate buffer system. So let's talk a bit about just the hormonal part that we just left a bit on the side for all this time. We have different let's say stimuli in different situations in the human body. We have the absence of loss of volume and high amount of volume. So in low amount of volume, we need to actually maintain the biggest amount of ions and water in our bloodstream in order to survive. So in this case, one of the very important mechanisms is the effects of the antidiuretic hormone. As the name suggests, antidiuretic means that it's against urination. So this is the logic and the mechanism in which the antidiuretic hormone works specifically in the kidney, is that it upregulates the amount of aquaporins that are present in these two segments of the distal DCT, and most importantly the collecting system. So the aquaporins in this case, as the name suggests, is pores that allow water to go from the tubule back to the human body. So this is one of the mechanisms that we save both, of course, water and as a consequence ions of sodium through the action of antidiuretic hormone. This is the one. Now the second one, the biggest system we'll be discussing all this time, is the renin angiotensin aldosterone system. So this is again a specific logic in which we have loss of blood, again loss of volume, not necessarily blood, loss of volume. So typically actually is found in the loss of blood very commonly. Now in this case is when we have the drop in the specific pressure, in the blood pressure, the macula densa will sense this drop of this specifically drop of pressure and activate the juxtaglomerular cells to produce renin. Now after renin is produced, we're going to have the conversion of angiotensinogen to angiotensin 1. Angiotensin, this is simply one of an easy name that will help you understand that one of the functions of the angiotensin 2 specifically is the constriction of vessels. Why? Because if you constrict the vessels, you shrink the lumen, and the same volume that is present in a smaller and let's say more constricted lumen increases the pressure of the present vessel. So the point is to maintain the blood pressure regardless of the volume. Of course, this is the first stage, just to maintain the blood pressure initially in the first stimulus. Now, the second part of this whole system will be the conversion of angiotensin 1 to angiotensin 2. Even though angiotensin 1 has some specific biological effects, angiotensin 2 is the one that activates and has this specific effect on both the vessels and, most importantly, the adrenal glands, where we'll have the production of aldosterone. Don't get confused, we'll discuss about the glands specifically in the hormonal system, in the endocrine system, but in this case just a short mentioning to fully comprehend the system and how it works. So this enzyme, the ACE, the angiotensin converting enzyme, is produced by the lungs, something you should have in your head, somewhere there in your head. So the first function and action of angiotensin 2 is to initially constrict the vessel to maintain the high amount of, to maintain actually the blood pressure. And the second is the production and secretion of aldosterone in the adrenal gland. As a consequence, what is actually aldosterone doing? Why do we care so much? Because aldosterone, in fact, increases the reabsorption of mostly sodium in the reabsorption of sodium. And as a consequence, because we have a higher amount of sodium in the area in between the interstitial area, then we're going to have the increased reabsorption of water. Because I told you before, water and ions should be considered as a unit. So this is the function of the renal aldosterone angiotensin system in very, very short synopsis, and just to get the whole idea of how the system works. So after this very long description of the function and structure of the kidney, let's actually visualize these structures in the microscope. So again, the easy way to understand where the cortex is and where the medulla is the presence of the renal corpuscles, and in simpler terms, simply the glomerulus. So if we zoom in the area that we can find these little balls practically present in the whole section, we should be able to locate the cortex of the renal lobe. So again, we can see the numerous sections right here, the numerous glomeruli present here. So let's zoom into one. So let's actually visualize this specific structure of the glomerulus. Again, this is the typical structure we saw before. Again, this is the vascular pole, and the tubular pole should be in the end right here. This whole segment, this whole part is the parietal layer of the Bowman's capsule. And the visceral, of course, is again composed by the podocytes. It's not easy to distinguish between the podocytes and the mesangial cells, but it's somewhat easy to identify the endothelial linked with the podocyte. For example, this simple squamous right here should be one typical endothelial cell. And now, surrounding this whole glomerulus, we should be able to identify the two different tubules that we discussed, the PCT and the DCT. The proximal convoluted tubule we discussed already, that has this pyramidally shaped. So this is again a very nice example of the PCT. Again, this is the second one. The DCT, the distal convoluted tubule, is always close to the also part of the macula densa. In fact, so we should be able to find it towards and close to the vascular pole. So this is the vascular pole again, and this is the ideal example of the distal convoluted tubule. Now it's very easy to see the differences in the nuclei, in the appearance of the nuclei, and of course the height of the cells themselves. We can see that the PCT has a bit taller cells, more columnar-like, short columnar to high cuboidal, and the typical DCT contains short cuboidal cells. So this is the first part we should discuss about and understand. Now let's actually try to find a collecting tubule, or try to find the actually, there it is, there we have it. This is a very nice example of the collecting tubule as it is. Again, we're going to find the very short, nice and typical columnar cuboidal cells, again short. We're going to find this is the here we're going to find the two different populations of the intercalated and the principal cells. Most of the population of these cells, of course, are the principal cells that react to this aldosterone and this whole structure of the aquaporins, and are the, let's say, the hormonally sensitive areas and affected areas in these cases. So exactly, this is the structure that we should find, and we should look it should look like as the collecting duct. So if we zoom out and again try to find the medulla, again, which is the area that does not contain not even one amount of glomerulus, glomeruli, we're gonna be able to see the two different tubules, the thin loops of Henle, and of course the other parts of the collecting duct. So in this segment we actually can see let's say more prominently, more ideally, these thin segments of the loop of Henle, exactly right here. And interspersed we're going to find other thicker ascending limbs of the loop of Henle, or possibly just a part of the collecting duct. It's not that easy in this case to distinguish the thick ascending limb with the collecting duct when in cross section. So let's talk about the ureter. Now before we talk about it, we should understand the whole process of production of urine. We already discussed the production of urine. Now we should talk about the drainage of urine. So the urine passes from the collecting ducts after, of course, it reabsorbs all the ions, water, substances, proteins, glucose molecules, other saccharide molecules that are being reabsorbed, and then pass on to the papillary duct of the renal pyramid, which is exactly the ending of the medullary part of the renal lobe. Now after this we're going to meet the minor calyx, which of course converges to form the major calyx in the end, and after that we're going to accumulate all the urine in the renal pelvis. After the renal pelvis is going to pass on straight to the ureter. So the ureter is nothing more than just a whole long duct that facilitates the passage of urine. So let's talk a bit about the structure of and the unique parts of the ureter. Now there is a very peculiar epithelium exactly in the ureter, also called urothelium, or also called transitional epithelium, transitional urothelium. Now this is the case in which we find stratified epithelium, and in fact we're going to find three layers of cells right here. first and always the basal layer, which of course as always is responsible for the mitotic rate of the epithelium. Then we're going to find the intermediate cells exactly in between, and the top are going to be called the umbrella cells, or also surface epithelium. What is the unique feature of this urothelium? Well, because urine itself is hyperosmotic, and any cell that is just a simple cell that is present in the hyperosmotic environment will end up being dead. So in order to prevent this whole changes in osmotic pressure and this whole negative aspects of the hypertonic solution when it comes to the, in comparison of course to the cellular fluid, the intercellular fluid, and the difference in the osmotic pressures. So the urothelium facility actually contains this unique structure, which is in fact a very thick lipid bilayer, and integrated in this thick lipid bilayer, there are the uroplakins, specific proteins that practically allow for the urothelium not to have any passage of water from the cell to the outer space of the hyperosmotic pressure, or the hyperosmotic liquid of the urine, and thus protects the shrinkage and ultimately the death of the surface epithelium. So this is the structure and the unique part of the urothelium. In fact, this is a very nice picture of the fluorescent microscopy that proves that this is the stratified structure with the umbrella-shaped cells and surface cells on the top. The middle ones, we're going to find the intermediate cells, and lastly the basal cells that are connected to the basal membrane. Exactly, this is how it looks like in the picture of the fluorescent microscopy. So let's visualize this in the microscope, shall we? This is the two sections of the ureter, the cross section and the longitudinal section of these structures themselves. So if we zoom in, let's go in the cross section of the traditional epithelium. Exactly in the top part, we're going to find this very peculiar appearance of this traditional epithelium, the surface epithelium. Now this is because these surface cells, the umbrella cells, are actually literally look like an umbrella, which is in this shape, and then the the connecting point to the other cells, to the intermediate cells. Of course, when you have a cell that is practically long and you cross section in the middle, you're going to see similar structures as this is in the picture right here. So this is exactly how we have the first epithelium, exactly in the urothelium itself. Now, exactly beneath, as always, you're going to find the lamina propria, which is again the very dense irregular connective tissue beneath it. Typically, we're should find a small amount of loose connective tissue exactly beneath the basement membrane right here, but here it's not really honestly that visible. Exactly beneath, we're going to find the muscularis externa. Now these are smooth muscles, of course, and as always have the two layers, the longitudinal, inner longitudinal, and outer circular layer. And in the upper part, we're going to have, because we're having increased necessity for constriction and increased necessity for of course pressure, we're going to have the triple layer, which is again long, circular, longitudinal. And last, we're going to find the adventitia. Now this is again the layers of the inner of the longitudinal section, the cross, and the outer part is going to be the lamina, the adventitia, which is of course as always sparse loose connective tissue along with adipose cells. You can easily see and clearly see in the picture right here, in the microscope right here. And this is pretty much the capillaries we can find surrounding the lamina, that adventitia, the tunica adventitia. So exactly the same picture we should find in the longitudinal section, exactly the same. And again, the typical appearance of the transitional, again the lamina propria, again the longitudinal section of the smooth muscles, the circular ones are in cross section, and lastly again the very short and sparse continuations of the smooth, again sparsely located, as you can see present, and lastly again the adventitia, exactly the same structure it was before in this cross section. So this is pretty much what we should talk about the ureter. There is not a structure that has a specific function other than just letting the urine pass by and reach the urinary bladder. Now, the common features with the bladder of the urinary bladder and the ureter is the epithelium itself. It's pretty much exactly the same. We're going to find again the surface umbrella cells, again the intermediate, and again the basal cells. The difference is here we're going to find the bladder in two different forms. Of course, one is going to be the relaxed and one should be the compressed. Why? Because when you have urine present in this bladder, this enclosed space of the bladder, then the incontinence of urine, of course, this urine will compress the structures and create this appearance of constricted or more compressed structure of the urothelium, and this is the final result of the fully let's say compressed structure of the urothelium. And this is again the more relaxed version, where we have no presence of urine exactly in the bladder. So one more feature we should discuss is the differences in the muscles. Of course, in this case, we should find again the muscularis externa, which actually, as you can see, there's a massively bigger difference in the amount of muscles we can find. Of course, the structure is exactly the same. Again, inner longitudinal, then a middle circular, and outer longitudinal. For example, this is where we find the first sections of the longitudinal smooth muscles. Exactly beneath in lower levels, we should find the middle, the cross section of the circular, as it is exactly right here. And last, we're going to find the outer long, which is exactly in the third and most outermost layer of the bladder itself. So it's pretty straightforward, pretty simple structure. And in the last part, we're going to find the serosa, and of course we can identify the serosa by the presence of the mesothelium. For example, this is again a very nice example of the mesothelium with a simple squamous epithelium right here present in the last section. Thank you very much.